Hello students, welcome back to another video lesson on chromosomal basis of inheritance. This is part 7. In this video lesson we will study about mutation, types of mutation and mutagenic agents. So this whole video is about mutation, right? Okay, so first, what is mutation? So before understanding mutation, you know that we evolve, right? So there is an evolution happen, and because of evolution different kinds of, you see those so much diverse animals, plants, bacteria and so on, living beings, right? And these living beings which are diverse is as a result of evolution. Now you can see today that, let's take an example for elephant, right? You see an Asiatic elephant and an African elephant. Those both elephants are having some morphological differences, and how these morphological differences comes because of some mutation happened in earlier ancestor. So mutation is a raw material, right, that promotes evolutionary changes, right, that promotes evolutionary changes. And mutation and recombination are the two major processes responsible for genetic variation, right? So we have genetic variation, that brings evolution, right? This brings evolution. And for genetic variation there are two processes involved. One is mutation, another is recombination, right? With the help of mutation and recombination, genetic variation comes and which promotes evolution. Okay, now what is mutation actually? So mutation is sudden change in the genetic material of an organism is called mutation. So sudden change in genetic material of an organism called mutation means, you know, that the DNA, the genetic material is a DNA. So there is some change in happens in DNA, right? Certain change happens in DNA because of which the whole outcome changes. Okay, that, and you also know that chromosomes bears the DNA, and or chromosome, condensed form of DNA is the chromosome. So the changes happens in DNA reflects its outcome, and because of which you see variations, and because of variations happens continuously brings about the evolution. Okay, the term mutation was introduced by Hugo de Veris. You know Hugo de Veris already. Hugo de Veris was one of the person who did the Mendel's experiment, right? So he gave the name mutation while he was studying on the evening plant or evening primrose known as Monothera lamarckiana, and proposed mutation theory. It is important for you who proposed mutation theory or who proposed the name mutation. It was Hugo de Veris, 1901. How he got the idea of mutation? So he was working on the plant, the evening primrose or Monothera lamarckiana, right? And he proposed mutation theory, right? Okay. There are two broad types of changes in genetic material. They are point mutation and chromosomal mutation. Okay, I want to tell you about this. I told you that DNA, so mutation is change of DNA. When you say mutation is change in DNA, right? Sudden change in DNA or genetic material. It's a sudden change in DNA. Now you already know that DNA is a linear strand, right? It's a linear strand, and if you condense them, they become chromosome, right? This is non-condensed form and this is condensed form. Uncondensed, right? Uncondensed, and this is condensed. Okay, so if some changes happens in the uncondensed form, okay, for example we have A, T, G, C. You already know that DNA is made up of nucleotides, so A, T, G, C, C, T, something like that. And suddenly some changes happens in one of the nucleotide. There are so many different changes can happen. You will see that. So some changes happen in one of the nucleotide that we will call as point mutation. It's called point mutation. Okay, if we talk about the chromosome, if something happens the segment of chromosome, there are so many things can happen. If this comes from here to here, or it may just skips out or deleted out, okay, those changes will be considered as changes happens in chromosome, known as chromosomal mutation. So this will be considered as point mutation, and this one as chromosomal mutation. So chromosomal mutation happens in the segment of a chromosome. Point mutation happens in one of the nucleotide of the DNA. Okay, so point mutation is smaller mutation, and chromosomal mutation happens or changes in more than one or two nucleotides. More than two nucleotides. Okay. Then mutational events that takes place within the gene are called gene mutations or point mutations, whereas the changes occurs in the structure or number of chromosomes is known as chromosomal mutation. Okay, so within the gene or within the sequence happens a point mutation, or if there's a change of structure. Okay, structure and number, two things are important, right? If the number increases, chromosome number increases or chromosome number decreases, that will be also considered as chromosomal mutations. And those agents which are responsible for the mutation are known as mutagens. Okay, so mutagens increases the rate of mutations. Mutation is a slow process, but mutagen increases the rate of mutation, right? Okay. Then mutations can occur either spontaneously itself, right, or induced. Spontaneously happens itself, and because of spontaneous mutation only evolution happened. Like we say man evolved from monkeys, so there was some spontaneous mutation happened, because of which certain monkeys becomes intelligent, large, change the body shape, brain size changes, right? So these things happen because of some spontaneous mutation. Induced mutation happens because we induce the mutation with the help of mutagens, right? And the production of mutants are the one which got mutated, right? Or which the mutation happens in which organism, that organism is known as mutants. Okay, through the exposure of mutagens. Mutagenesis cause பண்ணும். எதுல mutagenesis நடந்திருக்கோ அது mutants. சரியா? So என்ன ஒரு organism இருக்கு, ஒரு normal organism இருக்கு. இந்த organism நம்ம வந்து mutagen போடுறோம். mutagen போடுறதுனால mutagenesis நடக்குது. and in the normal organism mutant மாறிடுது. ரைட்டா? அதான் இதோட டேர்ம்ஸோட மீனிங். சரியா? So in a normal organism mutagen increase the rate of mutation. சரியா? So mutagenesis நடக்கும். mutation occur ஆகுது இல்லையா? occur ஆகிற process mutagenesis சொல்லுவோம். mutagenesis கொண்டு வர்றது mutagens, and mutagen நடந்துச்சுன்னா that organism is now a mutant organism. Okay, then major types of mutation. So first நம்ம பார்த்தோம், this is spontaneous mutation and an induced mutation. So a spontaneous mutation happens in the absence of mutagen. It happens suddenly itself, right? So அங்க வந்து mutagens தேவை கிடையாது, தானா நடக்கும். And induced சொல்லும்போது it happens because of the presence of mutagens, or because of mutagens induces mutagenesis, that's what it's a induced. சரியா? So organism இருக்கு, and then we have mutagens. That induces mutagenesis. And these organism becomes mutants. சரியா? இது வந்து induced ஆயிடுச்சு. induced mutation. Spontaneous mutation வந்து organism இருக்கும், and by some reason they become mutant. So இங்க mutagens கிடையாது, no mutagens. சரியா? ஓகே. Then based on cell type base பண்ணி somatic and germline, right? So somatic cells are non-reproductive cells or non-gametic cells, those cells which do not form gametes, okay, or body cells. So somatic cells la mutation nadandhuchina, they are somatic mutation, and those mutation occurs in non-reproductive cells, right? Liver cells, lung cells, kidney cells, any cells, right? So that we consider as somatic mutation. Germline mutation is the mutation that happens in the reproductive cells, you know, germinal epithelia, yeah, germinal epithelia. And they go gametogenesis, to make gametes. So mutation on the idhukku nadandhukonga, if some mutation happen, so in the gametes will be mutant gametes, right? Mutant gametes, and the mutant gametes la irundhu vara produce agra organisms irukilla, progeny will be mutants. So the changes happen in the germinal epithelial cells brings the changes in the progeny. அதாவது ஒரு ஜெனரேஷன் அடுத்த ஜெனரேஷனுக்கு போகும். ஜெர்ம் லைன்ல நடந்துச்சுன்னா ஒரு ஜெனரேஷன் அடுத்த ஜெனரேஷன் போகும். சொமாட்டிக்ல நடந்துச்சுன்னா ஒரு ஜெனரேஷன் அடுத்த ஜெனரேஷன் போகாது, அதே ஜெனரேஷன்ல தான் இருக்கும். ஃபார் யாருக்காச்சும் வந்து ஃபார் எக்ஸாம்பிள் வந்து லிவரில் ஒரு செல்லில் வந்து மியூட்டேஷன் நடந்திருக்குன்னா இட் ஸ்டேஸ் வித் தட் பர்சன் ஒன்லி. அது அவனோட குழந்தைக்கோ சரியா இல்லை பேரா குழந்தைக்கெல்லாம் போகாது, சன்ஸ் ஆர் கிராண்ட் சன்ஸ்க்கெல்லாம் போகாது. பட் இஃப் சேஞ்சஸ் ஹேப்பன் இன் ஜெர்ம் லைன் எபித்தீலியா, சரியா, ஜெர்ம் செல்ஸில் நடந்துச்சுன்னா when the person will have a baby, that baby will have the mutation, and that baby will transfer the mutation to his baby and so on. சரியா? ஸோ இது வந்து ஜென்ரேஷன் டு ஜென்ரேஷன் ஃபாலோ பண்ணும். Generation to generation. Same applies for plant also. Okay, megaspore mother cells, right? Or microspores, pollen forming cells or ovum forming cells nadandhuchina, they also brings the changes. So same thing applied for plant and animal, same system. Okay, then effect of mutation enna irkalam, loss of functions irkalam, சரியா? So loss of functions na ஒரு gene வந்து because of either deletion or addition of gene or nucleotide, they may lost the function, so function nadakama irkalam, right? And the function and the enzyme ude functions, for example it makes an enzyme, the enzyme ude function nadakaleena, that will bring some changes, சரியா? So loss of functions. Suppose there is a gene, ஒரு ஏன்னு ஒரு ஜீன் இருக்கு, அந்த ஜீன் வந்து என்சைம் ஏ செய்து வச்சுக்கோங்க, என்சைம் ஏ செய்து வச்சுக்கோங்க, and because of mutation there is gene A is mutated, so no A enzyme, so A enzyme mediated என்ன ஃபங்க்ஷன்ல இருக்கும், those will not happen. Metabolism depending on enzyme A will not take place. Okay, so இது ஒன்று இருக்கலாம். வேற reduce normal function இருக்கலாம். reduce normal function சொல்லும்போது the functional ability of an enzyme or a protein may be lost, right? So full-fledged of function பண்ணாது, பண்ணும், but rate of metabolic path will be slow-ஆ இருக்கலாம், சரியா? that we will consider as hypo என்ன low, சரியா? morphic mutation. So there will be reduced normal function எவ்வளோ பண்ணுமோ அவ்வளோ பண்ணாது, குறைவாக ஃபங்க்ஷன் பண்ணும். ஹைப்பர்மார்பிக், சரியா? அதிகமாக பண்ணும், ஹைப்பர் இல்லையா, அதிகமாக பண்ணும், எவ்வளோ தேவையோ அதுக்கு மேலே பண்ணும். that will also bring some changes, either that may be a positive change or may be a negative change. For example, just assume that there is some change happen in hemoglobin, and instead of carrying 4 oxygen molecule, it is carrying like 6 oxygen molecule. So என்ன ஆகும், so more oxygen இருக்கும், ATP அதிகமாக ஜென்ரேட் பண்ணுவோம், ஸ்ட்ரென்த் அதிகமாக இருக்கும், இல்லை மியூட்டேஷன், ரைட்டா? so it increases its ability. So irkalam. So mutations that increases an activity of a gene, so that we consider as hypermorphic mutation. Then gain of functions. Gain of functions if a gene is active at incorrect time or in inappropriate cells. Important, incorrect time and inappropriate cell. Example. For example, see, we have genes, all the genes in all the cells. That means genes from the 23 pairs of chromosomes. You already know that genes is DNA condensed form of DNA is chromosome, and chromosomes, 23 pairs of chromosomes are located in all cells. That means if we talk about insulin, so insulin is secreted by pancreatic beta cells, but the gene for pancreatic beta cells you can also find in a neuron, right, in a lung cell, in a heart cell, in a kidney cell. So these cells also have the gene that can make insulin, right? But insulin has to be made by pancreatic beta cells. Point 1. Point 2, insulin உடைய secretion or release எப்போ நடக்கும்னா when you feed, இல்லையா, நம்ம வந்து சாப்பிட்டு வந்து குளுக்கோஸ் ரைஸ் ஆகும்போது தான் அது ரிலீஸ் ஆகணும், ரைட்டா? அதுக்கு முன்னாடியோ பின்னாடியோ ரிலீஸ் ஆனால் வந்து யூஸ் ஆர் அதோட எஃபெக்ட் வந்து வித்தியாசமாக இருக்கும். ஃபார் எக்ஸாம்பிள் வந்து இஃப் ஃபர்ஸ்ட் நம்ம பார்ப்போம் இன்கரெக்ட் டைம், யூ ஆர் ஹங்க்ரி அண்ட் சடன்லி வந்து இன்சுலின் வந்து ஸ்பைக் அப் ஆகுது, வந்து ரிலீஸ் ஆகுது வச்சுக்கோங்க. என்ன ஆகும்? ஜென்ரலி வந்து மினிமம் பாடியில் பிளட் குளுக்கோஸ் வந்து 70 mg per deciliter இருக்கும். அண்ட் இஃப் இட் கோஸ் பிலோ தேட், ரைட்டா, இட் வில் கிரியேட் டெத் லைக் சிச்சுவேஷன், இல்லையா? டெத் லைக் சிச்சுவேஷன்னா குளுக்கோஸ் ஜீரோ ஆயிரும், இல்லையா? சடன்லி வந்து குளுக்கோஸ் வந்து கண்டினியூஸ்லி ப்ரொடியூஸ் ஆகிட்டே இருக்குது, குளுக்கோகான் இருக்குது, குளுக்கோகான் வந்து கிளைக்கோஜனை குளுக்கோஸை கன்வெர்ட் பண்ணிட்டு இருக்கும், அண்ட் குளுக்கோஸ் வந்து ஸ்டெடி ஸ்டேட்டில் வந்து வச்சுட்டு இருக்கும். பட் சடன்லி வந்து இன்சுலின் வந்து வித்தவுட் அ ரீசன் வந்து சடன்லி ரிலீஸ் ஆச்சுன்னா தோஸ் குளுக்கோஸ் வில் பி கன்சியூம், வில் கிரியேட் அ ஷாக். Point 1. Point 2 வந்து if insulin is produced by kidney cells, then think what about that? Our brain cells are producing insulin, right? So that will be a problem. So gain of function சொல்லும்போது if a gene is expressed in incorrect time and in inappropriate cells, that will be considered as gain of function. As you know that purine composed of A and G, and pyrimidine composed of C and T. When we talk about DNA, we are talking about DNA. So in pyrimidine, adenine, and uracil, right? So if a transition happens, okay, so G will be replaced by A or A will be replaced by G, vice versa, right? Or C will be replaced by T or T will be replaced by C. So let's see an example. If we have a sequence, for instance, A, T, G and C, okay, so if purine are getting substituted, then it will be like A, T, A, C, right? So here it was G was changed to A, right? So purine to purine, or let's take the same example, A, T, G and C. If it becomes A, T, G and T, for instance, so C is replaced by T. If such things happen, that will be considered as transition. Okay, let's see next. Next is transversion. In transversion, purine is replaced by pyrimidine, or pyrimidine is replaced to purine, right? So the thing is that, you know, purine again is A and G, and pyrimidine is C and T, right? So if A is replaced by T or G is replaced by C, that we will say as transversion. For example, we have again A, T, G and C, okay, and if it is changed to T, T and G and C, so you see there is a change, right? Purine becomes pyrimidine, or A, T, G and C can become A, T, G and G. So you see there's a change, right? So this will be called as transversion. Then insertion. Insertion is one or more extra nucleotide are added. Right, let's take the same example. A, T, G, C we have, and for instance here I added two nucleotide T, G, C. So see, we added two nucleotide, that will be insertion. So insertion increases the number of nucleotide. Or deletion. For example, again we have A, T, G and C. So for instance G is deleted, so it will be A, T and C. So G is gone. That will be also be a mutation, right? But how the mutation reflects the outcome, for that you have to understand about transcription and translation. So once you finish transcription translation, it will be more easier for you to understand this outcome. Then silent or synonymous. Synonymous there is a change, right? But the change didn't bring the change in amino acid, right? For example, let's see an example. We have a sequence A, T, G, C and A, again A, T, G, C. Okay, and now what happened? A, T, G, C and then A, say G, followed by same ATGC. Okay, so this AAA codes for lysine, right? And there is a nucleotide substitution. You can see is a transition where A is replaced by G, right? A is replaced by G. But interestingly, this sequence will be also coding lysine, right? So since the amino acid sequence is same, so there is no outcome because of this mutation. So this mutation is called as silent mutation. So here is written that no change in the amino acid encoded. So amino acid type encoded will not change, it will stay as same. Okay. Okay, then missense. Okay. When we have missense, then the meaning will change. Okay, sense is changed, that's why missense. Okay, for example, again we have ATGC, ATGC, and if we have CAC and again then ATGC, then let's again write ATGC followed by CAA and ATGC, right? CAA. So this one is substituted, right? Okay, so now what? CAC codes for histidine. It codes for histidine. However, CAA code for glycine. So you see the amino acid type of amino acid is changed because of a change in nucleotide. So such kind of mutation will be known as missense or non-synonymous mutation. There's a change in amino acid, as I given that this one original should be histidine, however mutated gives you glycine. Then nonsense. Okay, so nonsense mutation, what happens if we have a sequence like A, T, G and C, followed by U, G, U, followed by A, T, G and C, and a mutation happens which makes A, T, G, C into U, G, U, G, A, followed by same ATGC. Right, so this is original, this is mutated. Its original was making cysteine, amino acid. As you see that there is a change in nucleotide. Instead of U there is an A, and that's why it's a stop signal. So that will be say as nonsense or termination. So here the translation stops here. Okay, but to understand these things, this kind of thing, you should have a good knowledge on transcription and translation. Without knowing transcription and translation, it will be hard for you to understand the concept behind the types of mutation. Okay, so what I recommend you, when you finish molecular inheritance, then come back again and then go through it. That will be a good one, right? Okay, then frame shift. So in frame shift mutation, there is a change in the sequence of or the frame. For example, if we have a sequence ACG, ACG and ACG and then ACG and so on. Okay, so if we add or remove one nucleotide, for example, if you see here, yes, everything is ACG, ACG, ACG and ACG. Right? So if you add or remove a nucleotide, the whole story changes. For example, if I add one nucleotide, ACG, then I am adding one T here, T followed by again ACG, ACG and then ACG. Okay, so ACG code for an amino acid called threonine, threonine, and you know that the reading system is triple. So here is triple, fine, then it's changing, you see, it's changing. Right? So because of the change, this will be threonine, fine, but rest of the thing will be changing, and because of the change, the whole amino acid sequence will change. Right? That will be known as frame shift mutation. Okay, then there are two classes of gene mutation. Let's see. One is mutation that is affecting a single base pair, and the mutation that is altering the number of copies of small repeats, right, within the gene. Okay, so first is point mutation. So point mutation refers to alteration of single base pair of DNA or a small number of adjacent base pair, that will be known as point mutation. We already saw point mutation, right? Addition, deletion, frame shift mutation. Okay, so types of point mutation, here you can see that they categorize in two. One is base pair substitution and base pair insertion or deletion. So substitution we already saw, it may be purine substitution or purine pyrimidine substitution. Insertion or deletion may change the total number of nucleotide in a sequence, right? So here you can see it's a wild type, right, and they are producing an amino acid, right? So if there is a, say, transition or transversion, we see transition when purine is replaced by purine. For example, A is replaced by T or G is replaced by C. Okay, that will be transition. And transversion, if A is replaced by G or T is replaced by C, or purine is replaced by pyrimidine or pyrimidine replaced by purine, that will be known as transverse mutation. Okay, and then we have deletion or insertion or deletion, like insertion or deletion. Together we call them as indel mutation. Okay, then substitution mutation of indel mutation affects the translation because the number sequence will change as they are reading in triplet code. So insertion or deletion will change the sequence of triplet code, and because of which the amino sequence will change. Okay, then there is synonymous or silent mutation. We already saw that if there is a change of nucleotide but that nucleotide do not change the kind of amino acid, it is known as silent mutation. And non-synonymous or missense, if there is a change and that change brings the change in amino acid, that will be known as synonymous mutation. Okay, and nonsense mutation, if the mutation brings a stop codon, okay, that will be known as termination, redetermination, which is known as nonsense mutation. Frame shift mutation, if the frame shift, for example, like I show you here, it is given TGT then CGT. Okay, because of the change there is an addition, there is a change in the sequence, right? So that will be known as frame shift mutation. Then mutagenic agents next. Okay, mutagenic agents are those agents which causes mutation. So first time it was studied by Muller in 1927 in Drosophila using physical mutagen. Okay, so physical mutagens, for example, using temperature, radiation such as X-rays, gamma rays, alpha rays, beta rays, neutrons, cosmic rays, radioactive isotopes, ultraviolet rays are used as physical mutagens to produce mutation in various plants and animals. See, they are high energy, penetrable radiation, they can break the bonds, and because of which there may be insertion or deletion of nucleotide leading to a mutation. Then temperature, if you increase the temperature, the rate of mutation increases. While rising the temperature breaks the hydrogen bond. You know that they contain GC contain hydrogen bond, right? A and T contain double hydrogen bond, and G and C contain triple hydrogen bond. And if you raise the temperature, there are chances they will break out. And because of the breakout, a new pair will be formed that may lead to formation of mutation. Okay, that will affect replication and transcription. Then radiation. So radiation are classified into two types: ionizing radiation and non-ionizing radiation. So ionizing radiation are shorter wavelength and carry enough higher energy to ionize the electron from the atom. Example: X-ray, gamma ray, alpha ray, beta ray, cosmic rays, which breaks the chromosomes. And they causes mutation. Non-ionizing radiation, like such as UV rays, have longer wavelength and they carry lower amount of energy. So they have lower penetrating power than ionizing radiation. It is used to treat unicellular microorganisms, spores, pollen grains which possess nuclei located near the surface of membrane. Okay, then there's an example, for example, Saharbati sonara. Right? And Saharbati sonara is a mutant variety of wheat. They produced or developed by Mexican variety Sonara 64 by irradiating by gamma rays, and this was done by M.S. Swaminathan, who is known as father of Indian Green Revolution. Just remember for your exam. Then there is a castor aruna. It's a mutant variety of castor which was developed by treatment of seeds with thermal neutron in order to induce very early maturity. right? Like instead of 270 days, it was ready in 120 days. Then chemical mutagens. So chemical mutagens which induce mutations, or chemicals that induces mutations, are called chemical mutagens. So examples are mustard gas, nitrous acid, ethyl and methyl methane, sulfonate, right? Ethyl urethane, manganese salt, formaldehyde, eosin, anthracene. Okay, example given is nitrous oxide alter the nitrogenous bases of the DNA and disturbs the replication and transcription that leads to formation of incomplete and defective polypeptide during translation. Co-mutagens. So co-mutagens. Okay, the two. If you add something more, the rate of mutation increases and enhances. The compound which are not having own mutagenic property but can enhance the effect. You don't have, like, we have a compound which not causes mutation, but if you add with a mutagen, it increases capacity. For example, ascorbic acid increases the damage caused by hydrogen peroxide, or caffeine increases the toxicity caused by methotrexate. These are examples. Some examples given in the book, like mustard gas used as chemical weapon in World War I. Muller first time used X-ray to induce mutation in fruit fly. Stadler reported induced mutation in plant by using X-rays, and chemical mutagenesis was first reported by Orbach in 1944. Okay, so that much for today, but I strongly recommend you that you visit this chapter again after you finish transcription and translation, because lots of things you might not have understood because of lack of knowledge in molecular inheritance, right? So after you finish molecular inheritance, come back to this chapter and have a look again. Okay, with that I'm finishing this chapter. If you have any doubt, let me know. Have a great time. Bye bye.